in confined geometries, topology effects.
Yes.
Thank you, Giovanni.
Thank you, Sarah. So since everyone starts with a picture, we'll also start with a picture, you know, to thank our organizer and to see that proof that was really a funny conference. But I would like to thank, of course, the Newton Institute, University of Cambridge, for hosting me here and giving me this great time to, and all this possibility to meet people. So, what we are interested is in study, in practice, polymer physics of DNA. And so when you do polymer physics, then what you are interested, especially the critical exponents that explain how the size of the polymer grows as length of the DNA increases. We can have measure end-to-end distance distribution, measure correlation functions of DNA, and measure the shape also of the DNA. And this we are doing for linear, circular, and knotted DNA so that we can say something about the effects of topology. Today I will speak especially about circular DNA. So how the experiments are done? Well, this is DNA in solution, and in order to make images with the atomic force microscope, we have to deposit it on the surface so that it's there stable. And then we can, with our microscope, just go over the sample, take an image, and then we have the image of the DNA molecule that you see here. And then what we do is to actually trace the molecule, and then what I would say, once we have traced the molecule, we have the coordinates of the molecule, and then we know everything about the molecule. So then we are as good as any theoretician. We can calculate any functions that we want. We can invent whatever we want, and we can do it with our molecules. And here are just a few examples. This is linear DNA, this is circular DNA, and this is knotted DNA that we have recorded with the atomic force microscope. It has a very, very thin tip, and then we can scan the surface and see what happened. But there are different ways to deposit DNA. One, it's a strong trapping, means the surface is strongly positively charged and attracts strongly the DNA on the surface, or we can deposit it in a way that it can equilibrate in 2 dimensions. So the interaction is weak, and then as the DNA sits on the surface, it eliminates all the crossings and it spreads out on the surface. So these are the 2 ways we are studying, we can study DNA. And today I will speak about mostly about this system. Just to show you this, here on the right you see DNA is deposited in a weak way, so it can move on the surface. Each picture that you see is taken 1 minute apart, and then we had added an intercalant into the solution, and you see how the DNA supercoils positively. So it's really moving, it's alive on the surface. Here is attached in the strong way with a lot of charges, and the images are taken over half an hour. And if you look at this end of this molecule here, it doesn't move at all. So it goes down and doesn't move anymore. So these are the 2 ways, but we are, today I will just present mostly data about this situation where the DNA can relax in 2 dimensions. Just to present you the method, these are the images that we have. We trace with some program the molecules, and then what we do, for example, to calculate the end-to-end distance, we take a given contour length, calculate its end-to-end distance, move this window along the molecule, and then we can calculate the average end-to-end distance for a given contour length, and then you do this for all contour lengths. You do it for many, many molecules, and then you get out the end-to-end distance as a function of the contour length. As you can see, here is on a double logarithmic scale, and from the slope of this double logarithmic scale, because there is a power law relationship between the end-to-end distance and the contour length, we can get out the critical exponent. at the beginning, it will start with 1, and then it will go over to an exponent that, depending if it is in 3 dimension or in 2 dimension, it can be 0.58 in 3 dimension or 3/4 in 2 dimensions. But what is interesting about single molecule experiments is that we have all the molecules. So what we can do distributions. For example, each point that you see here is actually the average over a distribution. And then you can imagine if the molecule is rigid, there is no distribution, it's just a delta function. But if it is, as you can see here, but if it is flexible, then we will have a distribution like this. This is for semi-flexible regime. And if it is really perfectly flexible, then you get a distribution like this one. This you find it, for example, in Dejean's book. And we can measure this quantity, fit them, get out the critical exponents compared to the theory. And this is the advantage of single molecule experiments compared, for example, to scattering experiments or bulk solution experiments where you get out actually only average value of the quantity that you are measuring. So there are different quantities that we can measure. Is the radius of gyration of the molecule. You see here the coordinates of our molecule, and then we can calculate the radius of gyration, or we can calculate, especially for circular DNA, we can calculate the internal end-to-end distance as a function of the length here, or we can calculate the directional correlation function, which tells you how much the polymer is stiff and how it loses the memory of the direction as you go along the molecule. And from also what we can do, since it is a single molecule experiment, we can fit every molecule to a ellipse, and then we can calculate the shape parameters and get out the average shape of the molecule. And the predictions, I already mentioned them, is that the radius of gyration or the end-to-end distance, they scale with a power law with the length of the molecule, and the exponents are here. I just put them so that you remember here. The directional correlation function is an exponential function, and out of this exponential decay, you can get out the persistence length of the polymer. The shape parameters are here are just 2 limiting cases. If the 2 axes of the ellipse are equal, then you have a asphericity of 0. If it is a rod, we have an asphericity of 1, and polymers are just in between, and we can measure these quantities. So let's take now nicked circular DNA, nicked because we don't want effect due to supercoiling, and we deposit this on the surface as I showed you before. You see here 83 nanometers, circle 240, 900, 1500, and here 2,400, 2,000, 4,000, and 6,000 nanometer plasmid. You see already a big difference. When DNA is small, it looks about circular, and as it gets bigger and bigger, it gets, so to speak, flexible and flexible, more and more flexible. And now what we can do with this, we can do what I showed you before. We calculate the radius of gyration for these different plasmid lengths, and we can plot this in double logarithmic scale. And you see here for the small plasmid, a little bigger, and up to the maximum of 6,000 nanometers. And then you see immediately that there is not one single power law, but there are 2 power laws. One with nu equal 1, and one with nu equal 0.75. And what is the explanation of this? On short distances, DNA behaves rigid. It's like a rail track. It is on short distances, it's absolutely rigid, but the rail's direction in London is not the same as in Cambridge. So on long distances, it gets flexible, you know. And so the DNA goes from this shape here to this one. So it makes a transition from a stiff polymer, so rigid polymer, one-dimensional polymer, to the flexible polymer. And you see here exactly the change in critical exponent. Where it does this transition is exactly at the persistence length, because above this length, the polymer behaves more and more as a flexible polymer. Then for this circular DNA, we can also measure the internal end-to-end distance, and internal end-to-end distance as this shape here, because when you make one complete tour, you are back at the beginning, so it will have a shape like this, and then we can fit them with a very old equation, which doesn't mean that it's not good, and then we can get out the critical exponent, and you see that for short DNAs, the critical exponent is 1 because they are rigid, and as they get longer and longer, the exponent becomes 0.75, like for self-avoiding flexible polymer in 2 dimensions. Yeah. Now, what to say about the correlation function? The correlation function is defined like this. You take the tangent, and then you look the angle between the 2 points, and you calculate this average quantity, and it will actually decay exponentially. It will decay exponentially, especially on a range which is comparable to the persistence length. And you see here, for all the plasmid I showed you, they decay exponentially. The other 2 curves are for a persistence length of 40 until 60 nanometers. It means that DNAs really have a 50 nanometer persistence length. All the data lies between these 2 lines. But if you look on a larger length scale, on larger distances, well, the picture changes. Why it changes? Because when you start out with a circular polymer, then the vector is maybe in this direction here. But when we arrive at halfway along the molecule, the vector has to go in the other direction. I always say the molecule has to go home, so it has to change direction, and so it's negatively correlated in respect to the starting point. And so the correlation function is symmetric about the midpoint of the molecules, and then it goes back in a symmetrical way. Here is the correlation function unnormalized, and you see that something really interesting. At the midpoint, you see that for short molecules, DNAs, this one, the anti-correlation is maximal. And for long molecule, is less and less negative. And this comes by the fact that the long molecules are flexible, and so the midpoint is less anti-correlated in respect to the departure point. And so you see how the correlation just becomes less and less negative. And so this was studied by especially Sakaue and Wada from Kyoto University, and they made a function that takes this into account and explains the shape of this curve. And this shape of this curve is also typical for self-avoiding circular polymer in 2 dimension. If you look for the smallest one of the plasmid, you would expect a correlation function like for a circle, but it's not quite the case, because as you see here, we have a function, the dashed one is a correlation function for a perfect circle, and the data point don't really go through this. And if we take some shape which is slightly eccentric, then we can explain better the data. So it means that plasmid, short plasmid, they do not become perfectly circular, but are kind of elliptic, as we also saw this week. Short plasmid behave differently than an homogeneous material. Well, a little bit more about shape. These are the shapes for a short DNA ring, and these are for 2 microns. And what we did here, as you saw, we just took the molecule, oriented them all along the major and minor axis, so that they lie atop of each other. And you see that the short one, they behave like circles. The inner part is not explored by the molecule because it's stiff. But as you take longer and longer molecule, then the inner part of the ring, it is explored. But this what means? It means that the excluded volume interaction changes when you go from short DNA to long one. And this excluded volume interaction here has an effect on the shape of the polymer. And this was done, this numerical calculation was done by Erwin Frey at the University in Munich. And here you see the result. If you take a self-avoiding walk, as we have, you get the blue triangles. If you take a random walk, then you get the red one. And our data point go through the one for the self-avoiding walk. So we can see that the fact that they are circular and self-avoiding polymers, and the circularity increases the self-avoidance and has an effect on the shape of the DNA molecule. So now I will speak about a concentrated solution of circular nicked DNA, because concentrated solution somehow simulate confined molecules. In this case, the confinement comes from the other molecule, not because you put it in a microfluidic device or because you put it in a virus, but because the other are present. And so these are the images that we take. This is the experiment I showed you before that we will use as a reference. And these are the situation where we have the concentrated solution. And we use the PBR plasmid, which is about 1500 nanometers. And when you look closer to these images, you see that there are just a few types of molecules by chance. So there are the one that we were expecting, the one that are have the other around themselves, and then there are cases, because it's a high concentration, where inside one molecule we have another one. And so we have three cases. This is what we call the semi-dilute case, which is well known in polymer physics. And this is the case where we have a constrained molecule, in one case swollen by the inner pressure of the one that is inside, and the confined one, which feels the pressure of the other outside. And then we can analyze again this. We can take end-to-end distance, directional correlation functions, and like this, and we can study this molecule and see how they behave. So just by looking at them, you see if we take the semi-dilute one, it looks like it is more flexible than the other one, but DNA didn't change in between. So it's just how they look. And this is the effect of confinement. Effect of confinement makes looking the DNA as it would be more flexible. Persistence length remains the same, actually, but it looks like it is more flexible. Instead, the one that has the pressure from inside, it gives you the impression that it's stiff. So, you know, the effect is that the flexibilities, or the apparent flexibility, is influenced by the confinement of the molecule. And this is shown clearly when you take the correlation function and then you compare the correlation function with the one that you could simulate for circular molecule of different flexibility, you would get exactly this result here. Here you see our reference diluted circular DNA, and when it is collapsed, you see that the correlation function decays quicker because it looks more flexible. And if it is swollen, it decays slower because it looks stiffer. And you see it here clearly. Okay. Now, what happened with the molecule inside? Molecule inside, here are just two members of these categories. This one reminds you what you eat on the morning. It's a croissant. And we have tens of them. We can take the end-to-end distance if we want. We can take the correlation function, the directional correlation function, and calculate this quantity and see what we have. Look here, just the case for the directional correlation function. These are just simulations that were done by Guillaume Witz, who also did, by the way, all these experiments. And here is the correlation function. In red, you have the measured correlation function, and then the two dotted lines are for the two cases simulated here. One's when the confining geometry is a perfect circle, and one when it is another molecule. And this can explain the correlation function. And this correlation function, as you see, is very special. At the beginning, it will decay exponentially, but then it goes through a negative correlation about 1/4 along the molecule, and then it comes back again, and then it will do the exact symmetric on the other side. And why it becomes negative at 1/4? Well, this is typical for this croissant shape. So if you start here, your directional vector will look like this. When you arrive at 1/4, it will look mostly in this direction here, and so it will be anti-correlated. And then this anti-correlation is about lost. On average, it goes back a little bit, almost to 0, and then it will do exactly the same on the other side. And so this anti-correlation, this oscillating correlation function, is the signature of this croissant shape. And this croissant shape is the shape that has the lowest energy. If it would be a linear molecule, the lowest energy state of the molecule inside the circle would be a spiral, because this has the lowest elastic energy. But in the fact that they are circular, you cannot do a spiral, and then on the average, it is the croissant shape that is the most favorable configuration, and you see it in the correlation function of this system. Okay. So now I will add just something that was not in the in the title and in the abstract, is how topology and gene expression plays among each other, because we had a few talks about this, and then I would like to present what we did on this topic. And I always start this part in saying, well, that the naturing temperature of DNA is 70 degrees, life is at 37, so nature did a mistake. We cannot be alive, because we know we have to open DNA to express the genes, but since the denaturing temperature is 70 degrees, it cannot happen at 37, so we are all dead. You know, and where is the solution? Part of the solution, of course, because we saw how complex it is, gene expression control. Part of the explanation, it is in the topology. So you know that there is a linking number. The linking number is the twist plus the right of the molecule, and once that the DNA molecule is close, then this is a conserved quantity. I just show you this conservation of the linking number. We did many years ago an experiment. We took negatively supercoiled DNA from bacteria and add different amount of danorubicin, which is an intercalent. The intercalent just changes the twist of the molecule. It reduces the twist of the molecule, and because the linking number is constant, then the right has to increase. But since the right is negative, then it's increasing, it goes towards 0. And towards 0, you have a perfectly relaxed DNA, which you see here. There is no crossing left, so it's a perfectly relaxed DNA. And as you increase danorubicin, then it gets positively righted. It just supercoiled around on the other direction. And we saw this morning how the positive and negative supercoiling influences the action of the topoisomerases. And I don't know if the anti-cancer effect of these drugs here is to shift the effect of topoisomerases, because it creates actually positively supercoiled DNA. So I cannot tell if this is the case, but maybe the expert can give me some ideas about this. And so if you have a DNA, and this is the ideal form, it would have 8 twists, but you can underwound it might have 7 twists only, and there it starts the game of this molecule. So it could keep 7 only 7 turns, but it will not be happy. There would be torsional energy stored into the molecule, or it can relax this torsional energy into bending, and it will supercoil with a right of -1 and a twist of 8. Or it could release this torsional stress by opening a bubble, and in all these 3 cases, the linking number is constant. And it seems that in solution or in the cells, the game is between these 2 conformations here, as we also heard these last days. So what we did, we took a plasmid. Admit it's a pure topoisomers. It has a given linking number, a given deficiency in linking number, and then we deposit it on the surface. And what you see here, because there are fluctuations in solution, the plasmid can come down in all possible configurations. And some of the configurations have very few crossings, but you see here there are bubbles that open into the plasmid. And we did this with a lot of molecules. And then what we did, we measured actually the length of the bubble as a function of the remaining crossings. Why this is an interesting story is because if there are no crossings, then the right of this plasmid is 0. And so it has to, it puts all the deficient linking number into torsion and probably into opening of a bubble. But if the crossings are left, then it will be in the B form very happy and will not open any bubble. And so what we did, we looked at the whole plasmid. We measured some few hundreds of molecules, measured the bubble length as a function of the remaining crossings. Admit that it had a deficiency of linking number of 5, admit like the, or 10 or 20, whatever. If it has 0 crossings, it means it has a 0 right, but the twist or the DNA molecule must be unwound by 20. And so the bubble will correspond to 20 turns of the DNA molecule. But if I have 1 remaining crossing, then it has to unwind only 19 turns. So my bubble will be of 1 turn shorter. With 2 crossings, it will be 2 turns shorter, and so on and so forth. So the average bubble length just decreases linearly with the remaining crossings. And when you take the slope of this curve here, it corresponds exactly to 1 turn of double-stranded DNA open. So it's about 4.3 nanometers in single-stranded DNA length. And this is exactly the slope of this. So, but if we take in consideration only topology, it means that we only use this to calculate the bubble length, then the curve would be the red one here. And this curve here is actually the experimental one, is displaced by 12.5 nanometers. And Ralph Metzler did a theory about this, which is exactly based on the same principle that Craig Benham did. And it encompassed this theory, the fact that some of the torsion can be stored into the DNA. And so it is not all the missing linking number is not transformed into opening of the bubble, but part of it is stored into the torsion of the DNA. It was were the 3 cases I showed you before. And when you take parameters that are measured by other experiments, you see this is a constant, and you calculate this constant here, it makes exactly the 12.5 nanometers, and the curve goes through the point. So this is not a fitted line, it's just a calculated line with known parameters of other experiments. And so we can explain exactly what is happening into the system. And just to make it more visible this, I have here actually, unfortunately, a left-handed DNA, which has also as natural DNA, it has a weak point where then it will open, maybe a data box, something like this. And then when I put, when I take away a linking number, you see that it will just write up. You know, I can do it once, twice, and maybe 3 times, you know. It should have about 3 crossings because delta L K, it is 3. And then if it fluctuates in the solution, well, this right can disappear. This is a little bit difficult, but the 2 topology experts here of DNA can testify. You see, I have an opened bubble at this place here. This is a model of what happened in this case here. Okay, so maybe just a few minutes. So, half a minute. So, if we take a dimer of exactly this plasmid, you see that there are 2 bubbles forming here, because probably there are 2 weak points where it can open, and there it will open the system. Now I will just present you something kind of short about Topo 2. We heard this morning and the other days also beautiful biology, math, and experiments about Topo 2. And here is just Topo 2. What we did was to take knotted DNA. And what we wanted to do was to watch Topo 2 on action, saying, be able to watch how Topo 2 undo DNA. And for this, we took just a substrate. We put our DNA knot here. Everything it is in solution. And then we are doing imaging of this DNA in solution. It takes us about 1 minute to do the image. So it is not really fast. We saw this morning that in few milliseconds, Topo 2 can do its work. We are kind of slow. It seems that Swiss are slow. It's well known in Europe. And then what we have to do, if we want that the Topo 2 can act on the DNA must be loose on the surface. It must be free. Otherwise, if it is squeezed, like the molecule I showed you at the beginning, it will stay there. It cannot undo it. And so we change the condition of the solution, and what you will see is that DNA, it will float around, and the image will just be like blurred. It will look also that the DNA is broken, but the DNA is not broken. It's because that when the tip of the FM goes through, the molecule is not on the surface, so it doesn't appear. And then a little bit later, it is on the surface, and then it can be imaged. And then we add the Topo to the solution, and we observe the action. But it will be very quick, and too quick for Swiss people. Look here. So there are 2 knotted molecules. We change the condition. It's moving around. Topo 2 is added, and you see how the knot is open. It's just a few minutes, but at least we have a way to observe a little bit this, although with very, very little time resolution. And so maybe one day we have a super fast femtosecond atomic force microscope, and then I can do a cartoon movie of the Topo 2 acting on DNA knots. So with this, I would like to thank all the people that contributed to the work or part of the work I did here, especially Joseph Adamczyk, Guillaume Witz, and then I would also like to thank a lot the Isaac Newton Institute to having me here all this time and giving me this great opportunity to interact with the people here, and also especially Professor Moffat, Bayer, and Kephart for creating this beautiful environment. So my research was supported by the Swiss National Science Foundation and for the Carlsberg Foundation. And if you like my research, please drink Carlsberg beer. So thank you very much.
Questions, please. Yes, please.
Just a suggestion, because I'm not an expert in these things, but today or yesterday we were watching how one kind of topoisomerases was slower to make this entanglement or disentanglement phenomena, and there was another, I think it was topoisomerases 3 or 4, who was acting when the temperature was increasing or decreasing. And I was thinking, in your problem with two fats phenomena for visualize what is going on, why you cannot try with another kind of topoisomerases or changing the temperature to see if this process is slower?
Yes, this is for sure one possibility. What we did up to now was to take, for example, a topo 2 at maybe ADP or one ATP that blocks the action at a certain point, and then we could observe where it binds on the DNA and then do a very high resolution image. And what you're saying, for example, to raise the temperature with topo 1 it was, or topo 3, then maybe it will be slowed down. But possibly anyway, the strand passage is immediate almost for our time scales. But probably it could be a good way to have at least a picture before and after where the topo 2 sits, and then what happened afterwards. This is a good suggestion.
I missed the mono or divalent salt concentrations. Did you tell us that?
The divalent magnesium chloride was used to deposit weakly DNA on the surface. So the mica surface is charged negative. The divalent ions do the bridge and absorbs weakly the DNA. So we modulate the concentration of magnesium chloride so that it can spread on the surface. So this is the only use.
And monovalent is in the...
Yeah, we have monovalent. This, it depends. For example, for the topo 2, the last one here, we also modulate the monovalent, because we have to attach it, but not too much, and then we need this anyway for the action of the enzyme, and so. But
I meant in your curves, the ones where you had the split curves. You were looking at the flexibility and the conformation. Did you vary the salts for those experiments?
No, not no. We could not. We didn't measure the. So what you are saying, if we have measured the persistence length as a function of salt concentration, no, we didn't do it. We just kept always about physiological.
So like 150.
Yes, conditions. Yes.
Okay, Jason, one more.
So maybe a question and a appeal or something. And the question is, how big is this data set of curves, that you've captured? How big? How many curves do you have? How many of these DNA shapes?
We have a few hundred.
Oh, hundreds. Wow.
Yeah. No, we have automatic programs to trace the molecule.
Okay.
Otherwise, I need 10 PhDs and throw it away every end of the week to get the new set. So we have automatic tracing programs, or semi-automatic tracing programs.
So the appeal is, it would be an amazing thing for science if these curve shapes were somehow available. I mean, once they're done or ready or whatever, just to compare them to self-avoiding walks generated by other algorithms and see if there's anything different about the actual DNA shapes you've got versus this self-avoiding walk or that self-avoiding walk or what have you.
Yes, this, I mean, we of course, we if somebody asks us, we can give them, so it's not a problem for us. It would be even better because there are specialists, people that can do numeric stuff much better than we are doing, and so it would be a great help for us to have somebody analyzing them. Yes, thank you.
Cool.
That's brilliant. Well, shall we thank Giovanni
Thank
again?
you.